Have you ever wondered what the blueprint of life looks like? I'm not talking about DNA in its classic double helix shape, but something much, much bigger. Imagine taking all the DNA inside one of your cells, all 6 feet of it, and packaging it up neatly. What you'd get are these incredible structures called chromosomes. Today, we're diving into the fascinating world of karyotyping, the technique scientists use to take a picture of these chromosomes and see what secrets they hold. So, what exactly is a karyotype? Think of it like a family portrait, but for your chromosomes. It's a detailed study where scientists look at the structure and function of chromosomes. They do this through a really cool process. First, they need to get a sample of your cells. This could be from a blood test, amniotic fluid, or even a small tissue sample. The key is to catch these cells right when they are dividing, because that's when the chromosomes are most condensed and visible. Once they have the dividing cells, the real magic begins. They treat the cells with a special chemical to stop them mid-division, right at a stage called metaphase. This is like pressing pause at the perfect moment. Then, the cells are gently broken open to release the chromosomes, which are carefully spread out onto a microscope slide. But we're not done yet. To make them visible, the chromosomes are stained with a dye. This dye creates a unique banding pattern of light and dark stripes on each chromosome, kind of like a barcode. Now it's time for the main event: observation under a powerful microscope. A photograph is taken of this chromosomal spread. In the old days, scientists would literally print out the photo, cut out each individual chromosome with scissors, and arrange them into pairs. Thankfully, we now have sophisticated computer software that does this automatically. The software identifies each chromosome based on 3 key features: its size, its shape, and the position of its centromere. The centromere is that pinched-in waist you see on a chromosome, and its location, whether it's in the middle near one end, or somewhere in between, is a critical identifier. The final result is the karyotype itself, an organized profile of a person's chromosomes. Humans have 46 chromosomes in total, arranged in 23 pairs. The first 22 pairs are called autosomes and are numbered from 1 to 22 from largest to smallest. The final pair are the sex chromosomes. For females, this is 2 X chromosomes, or XX. For males, it's 1 X and 1 Y chromosome, or XY. So a typical human karyotype is written as 46 XX for females and 46 XY for males. But why do we do this? Karyotyping is an incredibly powerful diagnostic tool. By looking at this chromosomal map, geneticists can spot abnormalities. For example, they can see if there are too many or too few chromosomes, like in Down syndrome, which is caused by an extra copy of chromosome 21. They can also detect structural changes, like if a piece of one chromosome has broken off and attached to another. This information is vital for diagnosing genetic conditions, understanding causes of infertility, and even in some cancer diagnoses. It's a window into our genetic makeup, revealing the very foundation of who we are. So, from a simple cell to a detailed map of our genetic code, karyotyping is a cornerstone of modern genetics. It turns the invisible into the visible and helps answer some of the most fundamental questions about our health and biology. Thanks for joining me on this microscopic journey today. If you found this fascinating, make sure to hit that like button, subscribe for more science content, and let me know in the comments what topic you'd like to explore next. See you in the next video.